Hello everyone, I am Dr. Kamlesh Fadar and I am here to talk on gene therapy. As you all know that basically genes are a hereditary unit that consist of sequence of DNA which occupies a specific location on chromosome and determine a particular characteristic of a living organism. The process by which DNA is copied to RNA is known as transcription and then by which RNA is used to produce the protein is known as translation. Mutation of gene results in failure to synthesize a particular protein or synthesis of a defective protein. And when this mutation of gene occurs, it leads to the genetic disorder. Genetic disorder is a health problem caused by one or more abnormality in the genome. Gene therapy is a process that involves the insertion, alteration, or removal of gene within an individual cell and a biological tissue to treat the disease or disorder using a particular vector. The goal of any gene therapy is to treat genetic disease by repairing the damaged gene responsible for the disease. It involves introducing a normal copy of gene into the cell containing the damaged version. The cell can then produce a normal functional protein. Gene therapy is divided into two types, somatic gene therapy and the germline gene therapy. Cell other than the germline cell, that is oocytes and spermatocytes, are somatic cells. The somatic cells are not involved in the reproduction, hence it cannot be inherited. Somatic genes are thus non-reproductive in nature. Therapeutic genes transferred into the somatic cell, that is, example is introduction of gene into the bone marrow cell, blood cells or skin cells. Somatic cell gene therapy will not be inherited in the later generation. It is having a disadvantage that viral vector will combine with the cellular genome and thus infect the cell. This is known as the viral escape. Somatic gene therapy now can be broadly classified into the two categories, ex vivo and in vivo. Ex vivo gene therapy involves the transfer of gene in a culture cell which are then transplanted or injected into the patient. The ex vivo gene therapy can be applied to only the selected tissue whose cell can be cultured in the laboratory. It involves the following steps, that is isolation of cell with genetic defect from patient, growing the cell in the culture, introducing the therapeutic gene to correct the gene defect, selection of genetically corrected cell and grow, and then last is the transplantation or injection of that modified cell directly into the patient. It has the advantage of specificity of target cell, then safety and immunocompatibility, but disadvantage of invasiveness of cell grafting and risk of tumor formation. The direct delivery of gene into the cell of particular tissue into the patient body is generally called as the in vivo gene therapy. The corrected copy of gene is transferred into the body of patient. The in vivo gene transfer is necessary when the culture cell cannot be reimplanted in the patient effectively. But the major problem is inefficient targeting. Advantage is that it is a very simple one-step method and minimum invasiveness. And disadvantage of non-specificity of the target cell infection, and various different cell types can be infected when in vivo vectors are injected into the CNS including neurons, glia and vascular cell. And some in vivo vectors are toxic in nature. So this is a pictorial diagram of in vivo and ex vivo gene therapy. In situ gene therapy involves when gene therapy is done in restricted area or the particular site. It is called as in situ. In situ gene therapy requires the vector to be placed directly into the affected tissues or organ. The method is effectively used against the cystic fibrosis. The major concern of gene therapy protocol is the lack of efficient transduction of the targeted organ. Next is the germline gene therapy. It involves the reproductive cell, that is eggs and sperms. Gene therapies using the germline cell result in permanent changes that are passed down to the subsequent generation. But for safety, ethical and technical reason, it is not being attended at present. So we can distinguish between the somatic cell and germline cell, that somatic cells, when the therapeutic genes are transferred to the somatic cell, that is a non-reproductive cell, and when therapeutic genes are transferred to the reproductive cell, they are called as the germline therapy. Somatic cell line will not inherit it later in the generation part, but the germline, it is heritable and passed on to the later generation. Next is the approaches used in the gene therapy. There are the various approaches like gene modification and gene transformation. In gene modification method, that is a defective gene is modified to render it functionally normal. It includes the three sections. One is a gene replacement, gene correction, and gene augmentation. Mutated or non-functional copy of gene is replaced by the corrected normal functional gene in the gene replacement method. It is used for the disorders marked by the deficiency of enzyme or protein, but the disadvantage that it is very difficult process. Gene correction method involve only the defective portion of mutant gene which is altered to provide the functional gene, without precisely changing the gene back to the natural form. Advantage is feasible, but disadvantage that it is beyond the boundary of in vivo technology. Third, gene augmentation is aimed to supply the functional copy of the defective gene. Advantage that it is a very simple method to operate. So in nutshell, gene therapy approaches is correction of mutated gene, blocking the target gene expression, and last is insertion of new or corrected gene. There are various delivery methods for this gene, therapeutic gene. To transfer the desired gene into the target cell, a carrier is required, and the vehicle or carrier which is used to introduce this transgene is known as vector. It can be classified as viral vector and non-viral vector. In viral vector, it is again classified as integrating and non-integrating, including adenovirus, adeno-associated virus, retrovirus, herpes simplex virus, and lentiviral virus. In non-viral vector, it can be classified as a three type, that is called as the chemical method, physical method, and third is the biological method. In physical method, it is gene gun method, micro injection method, and magnetofection method. In case of chemical method, it includes the pure DNA construct, DNA molecular conjugates, lipoplex, and human artificial chromosomes. Regarding the vectors, we will discuss in next video. Now, regarding the advantages of gene therapy is that it has the potential to eliminate and prevent the hereditary diseases like cystic fibrosis and many more. It is possible to cure the heart diseases, AIDS, or cancer in the near future. It can be used to eradicate the disease from the future generation. But disadvantage that long-lasting therapy is not achieved by gene therapy due to the rapid dividing of cell. Benefits of gene therapy is short-lived. Immune response is the another disadvantage. Third is viruses which are used as vector, they may lead to the toxicity. And last is disorder caused by the defect in multiple genes cannot be treated effectively using the gene therapy. There are certain problems regarding the gene therapy such as regulatory approvals, immunity, inflammatory responses, target that one does not have the control over where the gene will inserted into the genome. It is very short-lived, and last is multi-gene disorder. So thank you so much.